Hello students. So now this is the biotechnology principles and processes. Okay. Now I'm going to start with artificial chromosomes as vector. That is BAC and YAC. BAC means bacterial artificial chromosomes and YAC which means yeast artificial chromosomes. Okay. So now artificial chromosomes as BAC and YACs. Okay. Now, in recent years, scientists have developed certain artificial DNA segments for transferring genetic material into eukaryotic cells. These artificial DNA segments are called artificial chromosomes. They are constructed from bacterial or yeast genomes and are accordingly known as bacterial and yeast artificial chromosomes. Okay, so now in recent years, scientists have developed certain artificial DNA segments for transferring genetic material into eukaryotic cells. These artificial DNA segments are called artificial chromosomes. Okay. They are constructed from bacterial or yeast genomes. They are constructed from bacterial and yeast genome and are accordingly known as bacterial artificial chromosomes or BAC and yeast artificial chromosomes or YACs. Okay, so now again what is bacterial chromosomes, artificial chromosomes or BACs or YACs. In recent years, scientists have developed certain artificial DNA segments for transferring genetic material into eukaryotic cells. Okay, for what? For transferring the genetic material into the eukaryotic cells. These artificial DNA segments are called artificial chromosomes. They are constructed from bacterial and yeast genome and are accordingly known as bacterial artificial chromosomes, that means BAC, and yeast artificial chromosomes or YACs. Okay. Now, YACs are constructed artificially in the laboratory by joining segments of original DNA of yeast and linear plasmid from bacteria. The telomeric, centromeric and origin of replication sequence are joined together to make a small but complete yeast chromosome. YACs are specially constructed or prepared for eukaryotic gene transfer and are found to be much more efficient than plasmid. Okay, so YACs are constructed artificially. YACs are constructed artificially in the laboratory by joining segments of original DNA of yeast and linear plasmid from bacteria and linear plasmid from bacteria. The telomeric, centromeric, the and origin of replication sequence and origin, the telomeric, centromeric and origin of replication sequences are joined together are joined together to make a small but complete yeast chromosome. To make a small but complete yeast chromosome. YACs are specially constructed or prepared for eukaryotic gene transfer, or prepared for eukaryotic gene transfer, and are found to be much more efficient than plasmids. Okay, so now this is the yolks and bats. Now next, is vectors for cloning genes in plants and animal cells. Vectors for cloning genes in plants and animal cells. Plasmids and phages are used as vectors for introducing foreign DNA in bacterial cells. Plasmids and phages are used as vectors for introducing foreign DNA in bacterial cells. The bacteria and viruses, especially retroviruses, are used to transfer gene or DNA segments to eukaryotic cells and transform them. Okay, the bacteria and virus especially retrovirus are used to transfer gene or DNA segments to eukaryotic cells and transform them. In nature, pathogenic bacteria and virus enter plant and animal cells and transfer their genes into host cells. In nature, pathogenic bacteria and viruses enter plant and animal cells and transfer their genes into host cells. They use protein synthesis machinery of the host cells. They use protein synthesis machinery of the host cells to synthesize their proteins to synthesize their proteins and thus force them to do what the bacteria and viruses want. For example, agrobacterium and retroviruses. So first now Agrobacterium tumefaciens. Agrobacterium. It is a pathogenic bacterium that causes crown gall disease in several dicot plants. Disease in several dicot plants. It delivers a DNA piece. It delivers a DNA piece to tDNA, piece of tDNA that transforms a normal plant cells into tumor cells, into tumor cells, and directs these tumor cells to produce chemicals required by the inducing plasmid, the TI plasmid. Required by the pathogen, and directs the these tumor cells to produce chemicals required by the pathogen. Chemicals required by the pathogens. The cells of tumor producing bacterium, the cells of the tumor producing bacterium have tumor inducing plasmid, producing bacterium, agrobacterium tumeficans faciens, have the tumor inducing plasmid. The TI plasmid, okay. This plasmid has now been modified into a cloning vector. This TI plasmid has now been modified into a cloning vector after removing tumor causing gene. So now this TI plasmid has now been modified into a cloning vector after removing tumor causing gene. Okay. In my next video, I will discuss about some another example like retrovirus, modified TI plasmid, etc. So now, bye students.